Hello everyone, my name is Tammy. I am presenting in Journal Club 3 and my topic is gut microbiome dysbiosis in rheumatoid arthritis patients post-treatment. So the article that I will be focusing on is titled Analysis of Gut Microbiota in Rheumatoid Arthritis Patients, Disease-Related Dysbiosis and Modifications Induced by Etanercept by Piccianti Diamanti and colleagues 2018. Here I have outlined the topics that we'll be going over in this presentation. So first we'll be going over the bottom line up front and the background, a little bit about the article objectives, the methodology, the results discussion, conclusion, and then questions that I have put together for discussion. First, let's address the bottom line up front. So why do we care? Firstly, rheumatoid arthritis is a known chronic autoimmune disease that causes the body to attack itself, specifically healthy joint and tissues. If untreated, this disease can cause deformities and disability due to the stiffness in the joints. So, advances in sequencing technology have allowed us to better characterize the gut microbiome. And now that we have this evidence available to us, there's growing data suggesting that gut microbiome dysbiosis is a key factor in immune-mediated inflammatory diseases. So it's kind of seen as like a hallmark for the diseases that I have listed here. So lupus, psoriasis, rheumatoid arthritis, and Sjogren's syndrome. There's many more, but these are the ones that I have cited in the references. So on to the purpose of the research. The authors in the study aim to look a little bit more into what the gut microbiota looks like in patients that suffer from rheumatoid arthritis after treatment. So this is actually one of the first studies to look at the effects of immunosuppressive drugs on the microbiome. And they are aware that the etiology of many of these inflammatory diseases are very unclear, so they know that we need to do further investigation in order to provide more targeted therapies for patients. So the research question, and I have it bolded here at the bottom, the authors want to ask, do immunosuppressive drugs have an effect on the composition of the gut microbiota in rheumatoid arthritis patients? So there is an unmet need for rheumatoid arthritis patients in this field of microbiome research. And on this slide, I have a timeline to depict when methotrexate was approved for the indication for rheumatoid arthritis patients, as well as etanercept, and when we really got started focusing on the human microbiome. So methotrexate and etanercept are commonly prescribed conventional therapies, and methotrexate was approved for 1947 on 1947, and sequencing technology has only been around recently. So we haven't had enough time to kind of look at the effect of the medicine on the gut microbiome, specifically for rheumatoid arthritis or RA patients. So only within the past couple decades or so has this gut microbiome space really taken off. Similarly, etanercept is the first tumor necrosis factor alpha inhibitor approved for use in RA patients in 1998. So I have here that the Human Microbiome Project, which was really the first time that we took a glimpse into the microbial diversity space in healthy humans, and we looked at if there's possible relationships between the gut microbiome and particular human diseases. So it's relatively new, and that's why this article is kind of the first in the space to do this. Since it is a new area for research, it's important because shedding light on the effect of immunosuppressive drugs on the gut microbiome can provide direction on disease management and potentially being able to improve alleviating symptoms of patients suffering with debilitating chronic autoimmune diseases. Moving on to the article. These were the objectives that the researchers wanted to address. So I have it broken up into 3 categories. The first category is analyze. So the researchers wanted to analyze the gut microbiota composition in untreated and treated rheumatoid arthritis patients. So they had multiple cohorts, which I will go over in the methodology slides, where patients were treated with either methotrexate, etanercept, or both, and these were 3 different categories. They wanted to compare. So they wanted to compare the gut microbiota composition in untreated patients and those receiving the treatments. And lastly, they wanted to investigate. They wanted to see how the gut microbiota composition related to the disease activity and immune function markers in rheumatoid arthritis patients. So when I go more into the methodology, there were specific clinical parameters that the researchers focused on when they were looking at the microbiota profiles. Methodology. So I split up the different methods to provide more information on the researcher's approach and how the study population was broken up, the sample collection and DNA extraction methods. They used bacterial 16S rRNA sequencing, and this data underwent bioinformatics analysis and statistical analysis. In this study, the patients were divided into 4 groups based on their current treatment. So out of 42 rheumatoid arthritis patients, 11 subjects were put into the treatment-naive cohort. So these were patients who never received immunosuppressive treatment before. 11 subjects receiving methotrexate were put in one cohort. 10 subjects receiving etanercept were also put in another cohort. And then there was 10 subjects who were receiving both therapies who were on the 4th cohort. The 5th cohort were the 10 subjects who were healthy. So I wanted to note here that patients and healthy controls who were taking steroids, non-steroidal anti-inflammatory drugs, antibiotics, or probiotics were excluded from the study, as well as, of course, if they had any other autoimmune diseases. So onto the sample and DNA extraction. On the day of collecting stool samples, there were disease activity markers, usually specifically chosen to see the activity of inflammation in rheumatoid arthritis patients. So DAS28, ESR, CRP, RF, ACPA, and dietary and smoking habits. Stool samples were collected and processed using the QIA-AMP DNA stool mini kit, and the DNA concentration and purity were assessed with nanodrop spectrophotometer. So for the sequencing, high throughput sequencing was conducted using the Illumina 16S metagenomic sequencing library preparation instructions. DNA extract was used to amplify the V3, B4 regions of the bacterial 16S rRNA gene. And the primers that were utilized, I have them starred, are listed below. So there's the forward primer and the reverse primer utilized. In terms of the bioinformatics, 16S metagenomics app was used to analyze the data from the sequencing, and the Shannon index for alpha diversity and species richness was also completed through the app. In terms of statistics, there was a correlation analysis performed using the Spearman rank, and of course, statistical analysis was performed using R. On to the results. So here we have figure 1, which is a Shannon diversity index of the filtered sequences depicting the species richness amongst the different groups. So the analysis of the Shannon index in each sample showed that there was no significant differences in the alpha diversity between treatment-naive rheumatoid arthritis patients and healthy controls. Additionally, there were no significant changes in the alpha diversity within each treatment group, which was the treatment-naive group, etanercept, methotrexate, or ETN plus MTX. These were compared to the patients who were not receiving any therapy. Similarly, there were no significant differences observed in species richness between these groups. So on to the next set of results in Figure 2. So here we see the varying profiles of microbiota in different treatment groups. Figure 2a is showing the phylum level, 2b shows the class level, and 2c shows the order level. So it's important to note that the authors only listed the top 8 classifications that resulted from the analysis. So although the distribution amongst the different groups remained the same at the phylum level, when looking at the treatment to control groups in 2A, there were actually striking differences in 2B and 2C. So in Figure 2B, there was a significant increase, 5-fold increase actually, in the class bacilli in rheumatoid arthritis patients when compared to healthy controls. And then in 2C, there was also a huge increase, a 17-fold increase in the abundance of lactobacillales in rheumatoid arthritis patients compared to the control group. Next, from this same data set, the researchers wanted to see if there was a difference in the composition of treatment-naive patients and the patients who received treatment. So, patients that took etanercept only saw a significant increase in cyanobacteria, denoted in 2A, and on that same path, there was also an increase in Nostosophistidae class and Nostocales order in Figure 2C, which are both belonging to the cyanobacteria phylum. There was a significant decrease in Delta Proteobacteria, which is a class listed in Figure 2B, and then Clostridiaceae family, depicted on the next slide. So that's going to be in 3A. So in terms of methotrexate, they saw that there was a decrease in Enterobacteriales, which was really the only significant alteration results. And then in the group with both therapies, etanercept and methotrexate, there was no significant changes observed in these patients. So on to Figure 3. This slide or figure shows the taxonomy comparing the abundance of family, which is 3A, genus 3B, and then in species, which is 3C. So although the text is very small on this slide, so I'm sorry about that, you might not be able to see it very well, but I'll point out the key findings from this picture. The figure breaks down the 16S rRNA sequencing data to the lower levels as in the previous slide. And again, the top 8 classifications were listed here. So there were also significant findings here. There was a decrease in the abundance of the genus Faecalibacterium, which is in Figure 3B, and its specific species, Faecalibacterium prausnitzii. So additionally, significant alterations were found in the genus Flavobacterium in Figure 3B, and then the species Blautia cocoides in Figure 3C. These were present in the control group, but were completely absent in the patients with untreated rheumatoid arthritis. So for this last figure, the researchers wanted to see if the composition of the microbiota was linked to the clinical parameters. And I have listed the clinical parameters. They were sex, age, disease duration, disease activity score on 28 joints, which is DAS 28, rheumatoid factor, which is RF, ACPA is anti-cyclic citrullinated peptide antibodies, erythrocyte sedimentation rate, ESR, and the C-reactive protein. So these are all clinical parameters that physicians use when assessing disease for rheumatoid arthritis. Additionally, lifestyle factors like diet and smoking were also taken into account in this Spearman correlation analysis. So this is actually the figure with the most information. So although I want to note that although not everything is known yet about the bacteria that were found in the correlation, it's nice to have this preliminary data to kind of pave the path for other studies to look at what these correlations really mean, right? So there is quite a bit of information, as I said, but I will point out the main findings that were listed on the paper. So disease duration was positively associated with Bacteroides cacae and negatively correlated with Parabacteroides myrtae. Disease or DAS score was directly correlated with Archaeota, gamma proteobacteria, Pastorellales, and Anaerobrancha savarsini. DAS score was inversely correlated with Erysipelotrichales, Coriobateriales, Coriobacteriaceae, Lactobacilliaceae, Collinsella, Bacteroides rodentium, and Collinsella aerofaciens. So rheumatoid factor and ACPA positively showed similar correlations with certain microbes. The positivity and the bacteria that were positively associated were Roseveria and negatively associated with bacilli, Lactobacillales, and Streptococcus vestibularis. ACPA positivity was indirectly correlated with Streptococcaceae, Erysipelotrichiaceae, Streptococcus, Bacteroides, Xylans solvens, and Lachnospira pectinoschiza. Erythrocyte sedimentation rate was directly correlated with Enterobacteriales, Roseveria faecis, and Streptococcus parasanguinus. Lastly, C-reactive protein was directly correlated with Parabacteroides distansonis. So as I mentioned before I went over these findings, it's interesting to look into what these bacteria are actually doing, depending on the increase and decrease in abundance. So on to discussion of these findings. So the key findings here, the authors noted that based on their findings and previous literature, the bacilli may play a role in pathogenesis since Lactobacilliaceae and Lactobacillus were more abundant in rheumatoid arthritis patients in comparison to healthy controls. They also noted that Faecalibacterium, which are bacteria that are known producers of short-chain fatty acid butyrate, that the reduction of these was observed in rheumatoid arthritis patients, and that it may play a role in the inflammatory process. So there is evidence to suggest that anti-tumor necrosis factor treatment, etanercept, is beneficial to the microbiota composition, because if we remember that we saw that increase in the cyanobacteria and in other different kinds of bacteria as well. So in terms of the limitations and strengths, a strength noted in the study was that it was a cross-sectional approach, which provided analysis of the gut microbiome on multiple clinical parameters for rheumatoid arthritis patients. And in terms of limitations, the authors acknowledged that the sample size was rather small. Granted, it was a pilot study, right? So it was kind of the first of its kind. And their opening remark was that the study was conducted on a smaller scale in terms of the study methods and procedures. So I think this can be viewed as a preliminary study which is exciting, right? Because more information is to come from this field. And one thing to keep in mind about this study is that it only looks at the gut bacteria in rheumatoid arthritis patients who haven't started treatment yet or are on etanercept or methotrexate therapy. So this kind of study design can tell us if there's a gut imbalance in these patients, but it won't exactly tell us, it can't tell us how to predict how rheumatoid arthritis will progress or affect the patient's overall outcome. So all in all, I really enjoyed this article, and I was particularly drawn into this topic because my mom has rheumatoid arthritis. She is currently on both therapies discussed in the study, etanercept and methotrexate, and she has been dealing with this illness for over a decade now, and it's quite debilitating. So seeing if we can learn more about how to manage this disease is very interesting to me. I actually told her that the fact that she's taking etanercept might increase her microbiome diversity, and she was very happy to hear that. So it might be beneficial for patients. So because this disease hits very close to home for me, I really look forward to more therapies and information to be discovered to improve the lives of patients. So to conclude our discussion about this study and the topic, I wanted to kind of sum up everything on the order of what we learned or what our background was and the outlook for the field. So that's kind of why I have this schematic for the conclusion slide. So the findings presented thus far further support existing evidence that there is a correlation between gut microbiota and rheumatoid arthritis. So it is known that rheumatoid arthritis is characterized by gut dysbiosis, and that microbiota likely play a significant role in the development and progression. Here in this study, we learned that anti-TNF alpha therapy, specifically etanercept, can partially restore a beneficial microbiota. Understanding the changes in gut microbiota will be critical to improving treatment strategies, and specifically developing personalized and supportive treatments. So these strategies can enhance the effectiveness and safety of care for rheumatoid arthritis patients. And lastly, of course, additional research in this area is very important to learn more about the etiology and the development of these inflammatory diseases. On this slide, I have some questions that I came up with that I think would be great discussion points for everyone. These might not have outright answers that are available in the research, but I would love to read into your hypotheses for these responses. So number one, how does the use of immunosuppressive drugs in the treatment of rheumatoid arthritis affect the composition and diversity of the gut microbiome? Number two, are there specific immunosuppressive drugs used in RA treatment that have a more pronounced impact on the gut microbiome compared to others? Number three, what are the potential mechanisms by which immunosuppressive drugs alter the gut microbiome in individuals with rheumatoid arthritis? Number four, can alterations in the gut microbiome caused by immunosuppressive drugs contribute to the development or progression of GI symptoms commonly experienced by RA patients? And number five, are there any strategies or interventions that can be implemented alongside immunosuppressive drug therapy to mitigate any negative effects on the gut microbiome in individuals with RA? Here are my references, and I would just like to thank you all for your time, and I look forward to reading your discussion posts. Thank you.